गुड मॉर्निंग टू एवरीवन। आज जो बात मैंने बोल रहा हूँ वो है सेंट्रल डॉगमा, दैट इज कॉल्ड सेंट्रल dogma. Okay, this is one of the part in your genetics, that is the central dogma. So what is the actual meaning of the central dogma? That central dogma, we know that is there is the three part is there. One is DNA, another is mRNA, and number three is the protein. These three elements are involved in this language of the central dogma. Okay, so where it come from central dogma? तो इस सेंट्रल डॉगमा कहाँ से आ रहा है? सेंट्रल डॉगमा क्या एक्चुअल क्या है? हम लोग जानते हैं जो द सेंट्रल डॉगमा of molecules of biology that state that the genetic information contain and the DNA. That means in the DNA there is the genetic information. Genetic information. Genetic information is there to the DNA. Okay, and the mRNA, in my previous lecture, that is the different types of the RNA is there. The mRNA, that means there is the messenger RNA. So that means messenger, that means the message, they give the transfer of the DNA genetic information to the protein. Okay, so central dogma, that's the genetic information contained on the DNA that is transferred to RNA molecules and then expressed in the structure of the synthesis of the protein. This is called the central dogma. Who coined the term? This is the pathway. So the Watson, Crick, the scientist, the Crick, they encountered the path. That is the path. That means DNA to mRNA, mRNA to protein. In 1958, they contain by encounter this path. This is the DNA to mRNA and to protein. This pathway is called the central dogma. Okay, this is the pathway. Okay, as we know that DNA, they have the genetic information. So this is the DNA, they contain the genetic information, and this DNA, they are double stranded. DNA are double stranded. Okay, there is some, this is A, T, C, G, A, T, the different types of this C, different types of the codon is there. So this is the sense stand, antisense, and this is the sense. This is a sense stand and antisense. This is a double stranded DNA. And the double stranded DNA, that's after the double stranded DNA, they are replicate. This is the replicate DNA, already replicate and produce two DNA. Okay, produce two DNA, two different types of the two DNA. Okay, this is the double stranded DNA. So this is after replication, they produce two different DNA. But here the DNA, after one process, this process is called transcription. They produce the mRNA. Okay, so the mRNA, that means they produce the RNA, one type of the RNA. So RNA is always single strand. The RNA is always single strand. So this is A, that they produce T. This is T, they produce A. This C produce G. This is G, C produce C, A, T. That is A and G. These are okay. And when they produce the RNA, this is the RNA, that is mRNA. Okay, this T produce A produce U. This RNA, they only change the T to U. Okay, this is the codon. This is G to C, C to G, T to A, and A to U, and G to C. This is the RNA. They are the single stranded. Okay, this is the single strand. And this single stranded RNA, and they after one of the process, this process is called translation. Okay, translation process, and they produce the protein. They produce protein. These are the protein. That means they are primary protein and secondary protein and the tertiary protein. This is protein synthesis. This protein, so DNA, they express their character by the protein. Okay, so DNA, the genetic information is their DNA, and after the protein, they express their character by the producing of the protein. So there is the ATG. Suppose this is G, it is C. So ATG, their three codon, they are produced within one of the three codon is there and produce one of the amino acid. This amino acid, these different types of three codon, these are produced three types of amino acid. These amino acid, they are produced the protein. So protein, we know the protein synthesis is there in ribosome. In ribosome, there is the two subunit. There are 50 subunit, 50S and the 30S subunit also. So 50S and 30S. After that, with the help of the tRNA and mRNA, the tRNA, mRNA and rRNA, they are produced the protein. Okay, this is all about this. That is the DNA. The DNA is carry the genetic information, and this DNA, one of the process, the transcription, then produce the RNA. This is the single stranded RNA. After that single stranded RNA, they produce the protein by the process of the translation. So there is the one process is the transcription and the second process is translation, and produce the protein. This protein are synthesis by the help of the ribosome. So this process is called central dogma. And Crick's, the scientist, they in 1958 they encountered the name of the central dogma. This is the pathway. It's called the central dogma. So transcription in eukaryotic, the DNA containing the store of information. Okay, and translation, the mRNA serve as a template in which the amino acid are assembled in the sequence of the necessary to protein to correct the protein. Okay, so they codon the carried by mRNA and the translated to in amino acid sequence and by the tRNA. The communicative internship between the mRNA and the nucleotide and amino acid is protein called the genetic code. So this is called the central dogma. That means central dogma is their process, the transcription and translation and produce the protein. This protein showing the character of all genetic information. There is the main character of the protein. Okay, thank you all.